All right. So this talk covers some of the things going on in my laboratory that are designed towards enabling the ultimate translation of extracellular vesicle-based therapeutics broadly for many different disease or injury applications. And I probably don't need to spend too much time on the background for this group about what extracellular vesicles are, which I'll call EVs through the rest of this presentation. But it's good to make sure we're operating from the same fundamental understanding. So EVs are cell-derived nanoscale particles that transfer information between cells in pretty much every biological system that they've been looked for in. And they come in many different varieties through many different biogenesis pathways. But we've traditionally grouped them into a couple of different categories in the field. So we have these EVs that bleb directly from the cell surface membrane that we've traditionally called microvesicles that maybe have a little bit larger size profile in the high hundreds of nanometers on average diameter. And then we have these EVs that are produced as part of the N-lysosomal pathway inside of cells from multivesicular bodies that have traditionally been called exosomes. Now, there's been a lot of discussion in the field about what to call these things, what's the proper nomenclature. Small EVs, large EVs have been proposed based on their size. I'm just going to call everything EVs. It's my own personal opinion that we lack the appropriate separation and characterization technologies to really determine very much about the populations of EVs that we have at the moment in the field. So we just have a very heterogeneous extracellular vesicle population in all of our studies. That's what I'm referring to when I'm talking about EVs. So I mentioned that goal of my lab is really to enable EV therapeutic technology. So why are EVs relevant for therapeutic development? Well, there's kind of two ways you can look at it. So one is that EVs are really like biological nanoparticles, biological drug delivery system. This was shown very nicely in some seminal work from Matthew Wood's group in 2011, where they reported in this paper in Nature Biotechnology that they could capture EVs, load them with cargo, load them, decorate them with a targeting ligand, and deliver them to a mouse and take advantage of some of the biological properties of the EVs, potentially able to cross biological barriers like the blood-brain barrier, and have some significant benefit to treat the brain in this case, where things like lipid and polymer nanoparticles and cell-based therapies have not been successful. So this was a very exciting paper, demonstrated a number of key concepts that you could really think about using EVs as drug delivery vehicles. That I read as a postdoc and really got me thinking about this field. In the same year, another paper from the Douglas Asordo's group showed that EVs really could account for much of the activity of circulating CD34 positive stem cells. And this was another different paradigm for EV therapeutic activity, where EVs are essentially the active ingredient in many cell-based therapies. And so, they have potential advantages over cells, which we'll delineate in just a minute. But really, these are two different things, but both of them very exciting and full of possibility. And so I remember reading these papers as a postdoc and getting really excited about this field, and end up getting to explore it as I became a faculty member a couple of years later. So you can use EVs for almost anything you can think of, anything you might be interested in. I'm pretty sure you could find some example of EVs being utilized in that application, right? So here's an anti-inflammatory and vascular repair, cartilage repair, angiogenesis, muscle repair, hair growth, spinal cord injury, anything you can think of, you can find EVs, being used for that. And so again, very exciting, but it does raise a question, how can that be the case? How can this, how can these EVs be useful for everything, every potential application you can imagine? The way I think about it is that, to me, where we are in the EV field is if you compared it to proteins. Imagine if all you knew about proteins is that they're different sizes and different cells make different ones. And all we can do is capture all the proteins a certain cell makes and apply them to a target cell and see what happens. That's kind of what we're doing with EVs at the moment. We're, and so you think about the gulf between what I just described and what an actual protein therapeutic is, right? You have to identify the specific protein that's causing the effect. You need to identify its target. You need to identify its mechanism of how it's working. You potentially need to enhance the potency of that protein. You need to potentially find a way to deliver it appropriately. All those things have to be overcome to make a therapeutic. And so that's kind of the mentality that I try to encourage my group to bring to this field, is that, we have a lot of interesting phenomena that have been reported. And now we're going to try to help bridge the gap from those very promising phenomena to where we can get to an actual therapeutic application at some point. So overall, looking at strengths and weaknesses of EVs for as therapeutics, they have some really compelling strengths. Their non-synthetic nature gives them potential biocompatibility advantages over things like lipid nanoparticles and polymer nanoparticles. Their safety profile gives them potential advantages over cell-based therapies, right? They can't divide uncontrollably. They can't differentiate into something nasty. They can't hide somewhere for years and years. They have a predictable clearance pathway. They have a predictable half-life. And so, in that respect, they have many advantages over cell-based therapies. And if they're having very similar activity, then you're going to get a real benefit there. You can also leverage things. You can leverage microbiology to design and control properties of EVs, which you can't do with synthetic drug delivery systems. You can leverage existing biological pathways, like the Wood group was kind of trying to do, where they took advantage of the properties of these EVs of being able to cross biological barriers. That's just a major design challenge for a drug delivery system that you no longer have to address because it's sort of already been figured out biologically. However, of course, there's certainly weaknesses. I mean, one of the weaknesses right now is just our limited mechanistic understanding. It prevents rational design of therapeutic strategies. And then there's some sort of manufacturing concerns. Can these be used off the shelf? Can they be targeted? Can they be made at large scale? Are they able to be made potent enough? And so we're kind of focused on this latter group of manufacturing, biomanufacturing issues as some of the main topics in our lab. So this is kind of our basic workflow, and sort of pie in the sky. We're not doing all this, or certainly not working in humans. But this is sort of the idea of how you could envision this working, is that you could harvest EVs from the source and modify them, put them back in a patient, or you could come up with a generic source like from induced pluripotent stem cells, or some other biocompatible source that could be used broadly for patients. But you're still going to need to do some things to make it probably a useful therapeutic. You're not going to just be able to go from okay, magic cell, source cell of desired to therapeutic without some more work being done in there. And so the things that we're focused on that I'll address in this talk are boxed in this figure here. Enhancing EV production and potency during the manufacturing process, loading EVs with cargo and controlling that process, and then how do you store EVs and enable their potential off-the-shelf use. So I'll address each one of these here and just show you a few things that my group has done in each of these areas. So we'll start with the cargo piece. And if we look at EV cargo, there's a lot of things that are attributed as EV cargo, right? And you can see this figure is an example. There's proteins, certainly there's lipids, and then there's various different types of nucleic acids. And you can see some information. MicroRNAs are generally described as being relatively abundant with respect to nucleic acids. DNA is rare. You can also find some long non-coding RNAs in there. And most papers would cite that either proteins or microRNAs are kind of what's really mediating most of the key biological effects of EVs. But the inclusion of microRNA especially is interesting, because if you take a whole population of EVs and you lyse them and analyze by PCR and look at the microRNAs there, you'll find lots of different microRNAs, but the counts will be very low. They'll be much less than 1 microRNA per vesicle on average. And so it indicates that either, they're sort of distributed into some subset of populations that's microRNA rich, or there, may not be that many microRNAs in any given EV. And it's just a sort of cumulative effect of delivery of many EVs to a cell that gives you the accumulation of microRNA that's needed to have any biological effect. So that may be how it works biologically. Of course, we don't know. We're still trying to figure this out as a field, but therapeutically, doesn't sound very promising strategy. You'd really like to have higher cargo loading to compete with sort of the types of technologies that would be utilized to deliver this type of cargo, such as lipid nanoparticles, and as we've all seen very fortuitously and gloriously with the mRNA vaccines from Pfizer and Moderna. So okay, how do we load cargo into EVs and enhance the cargo content to make them reasonable as drug delivery vehicles, as was envisioned in this original paper? The way this group did it was by electroporation. So this step right here, there's electroporation, enable the cargo to get inside the vesicle after it was isolated, and that's how the field was sort of launched. And so we and others followed along in the considerable footsteps of the Wood group on this. And we explored electroporation, in this case, for looking at DNA loading. And you could find some interesting things. So, over here, the main thing we show is, yeah, you can load a lot of linearized DNA constructs per vesicle. You all of a sudden you get it from less than 1 to hundreds per vesicle on average. So that's certainly very useful. It's a diffusion mediated process of loading, so there's some size limitation, which isn't surprising. We found it to be between 750 and 1,000 base pairs. And then there's things like if you have a bigger vesicles, like this population, which we called microvesicles in this paper, you can load more DNA into bigger vesicles. Right, that makes sense as well. But interestingly, you see this effect here as well, that if you load in DNA and then you treat with DNase, if you don't have DNase, you detect a lot of DNA still. But then if you treat that with DNA, so much of that, what you detect is not protected from degradation. So it's not truly encapsulated or incorporated in EV in such a way that it's protected from nuclease degradation. And we found that to be really the result of a lot of aggregation of cargo. And the aggregation also really limited the functional effects that we were able to get from this cargo. We really were able to get very minimal gene transfer, gene expression as a result of this DNA delivery. And it was very difficult to deliver any amount of plasmid DNA, which certainly was problematic in terms of gene expression as well. So in our hands, clearly there's some benefit, and you look at small stuff. I mean, with this being an order of magnitude 10-fold higher than your typical siRNA or microRNA. So, you can clearly see that, you'd be able to load a lot of those things, of small things in there, and, maybe there'd be less aggregation. Some people in other groups have investigated this and found similar results, but in our hands, electroporation wasn't working the way that we hoped, causing some problems with the cargo. And so we wanted to explore some alternatives. And one of the first ones we looked at was sonoperation, which is a similar concept, except instead of using electric field to make holes in the vesicles to allow cargo to go in, you're using sonic energy to accomplish the same task. We saw that you can have relatively good stability of the proteins in the presence of sonication of the EV-associated proteins, is what this data shows. And then most importantly, we had much, much less aggregation of siRNA, in this case, with the sonication method as opposed to electroporation method. So we measured aggregates greater than 300 kilodaltons and saw many fewer of them in the sonication method. But you still get degradation. If you see here, as you get up to 30 seconds and 60 seconds, you start to see degradation, and sonicate for 300 seconds, you just, completely degrade the cargo. So, nice benefit here, but still some issue with the labile nature of nucleic acids with this approach. But we can load lots of different things, siRNA, microRNA, single-stranded DNA, and we demonstrate that you can retain function of something like siRNA, knocking down mRNA and protein expression of an oncogene HER2 here in breast cancer cells, as this was measured. So, and then another benefit is that if you look at something like an MSC, mesenchymal stromal cell derived EV, which are used for many different potential regenerative applications, you see that sonoperation does not negatively affect the sort of endogenous EV activity of these. This is a gap closure assay, which I'll show several other slides of, where we use a machine to make a gap in an endothelial cell monolayer and measure how quickly the gap is restored based on exposure to media with a bunch of growth factors, media without growth factors as the controls, and then adding in the EVs, in this case MSC EVs, from basically just not disturbed and sonicated types. You get some activity with those. So this is like kind of your positive control, and it's very similar when we sonicate. So that's good to see. And the activity is retained, which is crucial. So to summarize this piece, we can reduce aggregation with sonoperation relative to electroporation. You can preserve the activity and the integrity of the EVs, but you still have this risk of degradation because you're adding a lot of energy to the system, and you just really can't avoid that. So we wanted to see, okay, is there a way to get cargo in EVs without adding so much energy to the system? And so we tried this scheme of protonation, where you follow this process where you sequentially dehydrate and rehydrate EVs to create a pH gradient, where you then have electrostatic-based interactions of EVs with the cargo, and that facilitates the loading. So this worked very nicely in terms of increasing the actual amounts of EVs, which we calculated would constitute almost 70% of the internal volume. So couldn't hope to achieve very much higher loading levels than this. We had the same versatility of cargo loading with respect to microRNA, siRNA, DNA, and then we see that excess cargo is not degraded after loading, which is the point of using this process. The change of EVs in this way, we worry that it might cause the EVs to become more immunogenic by kind of getting rid of all the glycosylation or any of the proteins on the surface, but it didn't seem to have much of an effect. We didn't see any difference in immune cell presence in blood after multiple IP injections of these EVs. And we were able to demonstrate, biological effects, the same type of stuff that you could achieve with other loading methods where, you could knock down a housekeeping gene, you could demonstrate a dose-dependent response of cytokine secretion, and you could, again, dose the anti-inflammatory effect of this miR-146, which, is just associated with the anti-inflammatory effects, and we show that knocks down IRAK1 expression, it suppresses IL-6 secretion. So, bottom line is we can get nice functional delivery of microRNA cargo with these protonated modified EVs. And this was work that was we were assisted with by Professors Lin Zhao and Wei Chao at University of Maryland, Baltimore, who've been our collaborators for several years and really, valuable contributions to this project. So for the protonation method, now we can load cargo without applying external energy. So you remove the degradation part of it. But you still don't have necessarily a great scalability with this approach. And that's something that actually plagues all of the different loading approaches that we've discussed. So if I can summarize this part overall, we've looked at several different loading methods in our group. Electroporation has limited utility in our hands, but it's still utilized by many groups. And, you can see many big papers coming out with electroporation as the loading method. So, it is what it is. Sonoperation and protonation are alternatives, very useful, but each with their own limitations. All of these methods, are concerned with respect to scalability, and may not really be as useful translationally as approaches that, are allowing you to modify the parent cells to produce EVs with certain properties, cargo, enhancements. Unfortunately, just like, straightforward transfection to overexpress like a given microRNA, doesn't always really work out to where then you get EVs with that, high levels of that RNA. And that's something that, many people, including our group, are working to understand better. Okay, so that was some of the work that we've done in the loading arena. I want to tell you just briefly, about some less sexy but yet important work about understanding how to best store EVs to enable their potential off-the-shelf use. And certainly we know that any sort of cell-derived therapy has an opportunity to be very expensive, and that's limiting its applications and limiting its utility even in the United States or Europe, much less low and middle income countries. And so, not necessarily achieving equity of treatment, which is something, that should always be a goal of any therapeutic. So, having it sort of so-called off the shelf would certainly, change the profile of EVs as a therapeutic platform, therapeutic technology. So we've investigated some storage conditions using both freezing and lyophilization, and just come to some interesting results that I want to go over here. So both freezing and lyophilization enable the maintenance of bioactivity of EVs. And we measure this a few different ways. Here's the gap closure assay again, where you have your positive controls up here, your negative controls here, and then your EV control here, these freshly isolated EVs, MSC EVs in this case, give you this amount of gap closure. If you store them at room temperature at 4C, you really see a big drop off. If you freeze them and either -20 or -80, or if you lyophilize them, you see a nice retained activity, very similar to the freshly isolated EVs. And then with respect to the frozen ones, even if you thaw them, we see this same retain. These are the same data here. But even if you freeze and thaw them several times, 5 times in this case, you still see a nice, you still see some activity, maybe a little bit of drop off, but it does indicate that you have some leeway there, and it's not totally going away when you freeze it. Here's another demonstration with activity, anti-inflammatory activities. Again, it's IL-6 that I mentioned previously. So in this case, we have macrophage cells that we stimulate with LPS, TLR4 ligand that stimulates IL-6 secretion. Dexamethasone is an anti-inflammatory control molecule. And then we have fresh MSC EVs, give you a nice, preventative effect here. When you store them at room temperature or 4C, nothing. But when you freeze them, you get some benefit, and then lyophilization, it looks very similar to the fresh EVs. And again here, when you freeze thaw, we see the same retained activity, which is great to see. So both of these are working pretty well in this case of enabling the same types of activities, at least from MSC EVs, which we measured in this case, and which are a key category of EVs that are being developed for many different applications. What about with loaded cargo? So we just finished talking about cargo loading. How do freezing and lyophilization affect that? Here, the story is a little more complicated. So if we look at our freshly isolated EVs and we normalize that to 1, when we store at room temp and 4C, it's almost all gone. But even when we freeze and lyophilize, we see a significant drop. You're only retaining about 20% of the loaded cargo as compared to the freshly isolated EVs. So that's significant, and we're still trying to understand where it goes and why it comes out. But interestingly, you get you still get a biological effect that you might expect. So here's the same IL-6 assay. Here's the effect of the fresh EVs in this one. When we add the anti-inflammatory miR-146, it improves it over the fresh EVs. But even when we're storing it at room temperature, so maybe that cargo goes away, but you still are maintaining the effect of the MSCs in this particular assay. And in all the conditions, you still get some significant benefit. So it may be the case that even though we're losing a lot of cargo, you're still going from hundreds or thousands of EVs or of copies of microRNA per EV to hundreds, and that's still a lot. That might be sufficient to give you the benefit of the biological effect, even though it's not as highly loaded as it was, as it would be if it were used fresh. Finally, we've shown that lyophilization in particular enables retention of loaded cargo, in this case, a long non-coding RNA called hot air in MSC EVs in an in vivo model of wound healing. And you see here that you get a little bit of benefit from these hot air loaded MSC EVs compared to control, and the lyophilized versions work very similarly to the freshly prepared hot air expressing MSC EVs. So these are all good signs, and trying to answer this question: can EVs be used off the shelf? They certainly can be frozen and thawed and retain their endogenous activity, although we may see significant losses in their loaded cargo. Lyophilization, however, does seem to be able to be really promising for this and be useful for really making EVs potential off-the-shelf type of therapeutics. And so that's really exciting, and again, something that would be a real advantage over a lot of competing therapeutic modalities if it really bears out that that's able to be done broadly. So yeah, something exciting from our end to observe. Okay. So the last part of this that I want to focus on is here, where we're addressing different production, enhancing production, and also conditioning cells to enhance the potency in the EV production process. Excuse me. And if we want to achieve scalable EV biomanufacturing, we can take some lessons from cell therapy because there's sort of similar challenges involved. EVs, however, are less dynamic than cells, which is an advantage, right? There's less ways that they can change than cells can, but there's extra steps. There's more potential for contamination. It's more difficult to work with smaller things. So there's a trade-off there. Most of the current knowledge about EVs in the field is generated from studies, using EVs that are generated by cells grown in conventional tissue culture and isolated by ultracentrifugation. And neither one of these technologies is really readily scalable, for large scale use in humans. It's going to be a real challenge to take those technologies into a bigger scale. So there has been work in developing bioreactors, and specifically hollow fiber bioreactors been utilized for EV production. And these are now available commercially from several companies. It's a very useful technology. If you have cells that are compatible with growth in the hollow fiber cartridge, this is a great way to produce a lot of EVs very quickly. The limitation, though, is that there's not very many knobs to turn. There's no potential for, controlling the environment in a very specific way to potentially enhance the properties of the EVs. And that's important because some of the things that we've observed in our lab are that, the parameters of cell culture are really important in the EVs that end up getting produced. If we look at even something as simple as cell passage in MSC EVs, we look at that wound closure assay, we really see a drop off between, early passage and later passage. If we look at even the density of the cells that are seated in a flask, we see a difference in the amount of EVs that are produced, with lower densities producing more EVs per cell than higher densities. And so, we would love to be able to have as much control as possible over the EV production environment, so that the more we learn about different effects of what impacts EV production and function, we can integrate that into the design of the bioreactor, and get a superior EV product at the end of the day. And so we have partnered with an expert in this area, John Fisher, in my department at the University of Maryland, and have been utilizing 3D printed perfusion bioreactors for EV production. And so here's here's just how it looks in the hood, and here's some cross sections and basic information about some of the scaffolds that we've been using. But we can dramatically increase the number of EVs that are produced in the same way that hollow fiber bioreactors have shown. But the nice thing about this is that we're using 3D printed cell culture chambers, and so we have very reproducible, very finite control over the properties of those chambers. And as we learn more about how the bioreactor works, we can modify those in a reproducible and scalable manner, which is something that there's not really any other option to do right now. So we first demonstrated the utility of this approach looking at endothelial cells. We use this type of cylindrical tiered pillar scaffold and just connected to a pump with media, where we can project what shear stresses and flows are going on through the scaffold, make sure that the cells are alive, growing in around the pillars as expected. And we were actually following up on work that we previously did in endothelial cell derived EVs, where we demonstrated that exposure to low concentrations of ethanol actually drove the increase in gap closure activity, which we found to be associated with a couple of long non-coding RNAs, hot air and mallet1. And so this is a very busy slide, but I'll walk through it. So what we were comparing in this experiment is EVs generated in conventional culture flasks and in the bioreactor. And then we also grew them in the scaffold without flow, just to kind of demonstrate what the role of flow might be. And so if we look at production, and we just compare flask to bioreactor, it's clear, and scaffold, there's nothing special about like growing them on this little piece here in terms of like really dramatically enhancing their production. When you put them on their flow, that's when you see the dramatic increase in the number of EVs produced. There's a couple of different explanations why that might be that we're currently pursuing and trying to understand more in the lab. Then we look at the activity again in the gap closure assay. We have our positive and negative controls here. We have a little bit of activity with the normal endothelial EVs, but then when we expose them to ethanol, we get this increased activity, and that's especially apparent in the bioreactor. So we were worried that, if we're making a lot more EVs in the bioreactor, is their activity actually diluted? But when we, load a normalized amount, the same amount of EVs, in this experiment, we see that the activity of the bioreactor EVs is as good, if not better, than the flask EVs, which is great to see, and something that we certainly want to understand what specifically is driving that, and that's one of the current focal areas in the lab. And then interestingly, what we also saw is that these long non-coding RNAs, hot air and mallet1, were both still overexpressed in the bioreactor, same as we saw in the original flask culture experiments. And so important takeaway from this is that we can use this bioreactor, we can increase production of EVs and maintain or even improve their activity, and also understand that it may still be at least partially by the same mechanism. And so that's really important, because that means that something that we understood from a 2D culture environment, we can actually transfer to this bioreactor and still, potentially maintain that understanding, or not just sort of starting from scratch of understanding how this works. So it's a really important finding for our group to know that, and still be able to leverage all the work that the whole field's done from conventional flask culture-based EV generation. And I already showed you before that this hot air long non-coding RNA is useful cargo in vivo in MSC EVs, as it significantly improves wound repair in a diabetic mouse model. Okay, so the summary of this part is that hollow fiber bioreactors are commercially available now, and so if you have a sort of golden EV producer cell that you're happy with, you can get a hollow fiber bioreactor from a few different companies, and if your cells grow in there, you're good. You're going to produce a lot of EVs. where we feel like the perfusion bioreactor technology fits in is, it's competitive in terms of the production, but it gives you this ability to really modulate the um, properties of the scaffold to potentially, mechanically program UVs to have certain properties. And we have a lot of work ongoing that's unpublished that I can't talk about here, but a lot of exciting follow-up in this area that'll be coming out in publications soon. Okay, so if we go back to the overall summary, advantages and limitations of UV therapeutics. Our goal as a lab has really been to address some of these weaknesses and try to bridge the gap between, enabling us to really maximize some of the strengths of this approach. So we mentioned the off-the-shelf capability, that seems like it's potentially solvable through conventional means without too much, new solutions having to be found. We also looked at potency with respect to drug loading. There's some different options there, some strengths and weaknesses to all of them. We're also in our lab exploring the different ways of manipulating parent cells and trying to understand what's the best approach there and how they really compare in terms of loading. But there's certainly something that hopefully we can come to as a consensus in the field of, what's the best way to get cargo in these EVs in a way that's scalable, reproducible, and applicable to many different cargo molecules. And then biomanufacturing, we're hopeful that the perfusion bioreactor approach can, as we improve other things like our mechanistic understanding, the perfusion bioreactor approach really allows for leveraging of that to feed back and maybe even improve potency and production even more. So we're really excited about the potential of that approach. So I certainly need to highlight the work, highlight the people who did all this actual work while I sat and typed at my computer. The members of our lab are shown here on the left, and specifically the work I talked about today was from Tekla Amachane, David Patel, Anjana Jairam, Louis Bourne, and Daniel Levy, who I mistakenly forgot to highlight there, as well as Steph Kronstadt, who's contributed in some of the bioreactor stuff. Ellie Potash is working on some of the loading work as well, following up from these things. We've also had really, incredible input from some key collaborators, John Fisher, John Harmon at Johns Hopkins with the animal studies, Wei Chao at the University of Maryland, Baltimore, from the inflammation work, and some of the other folks that we didn't necessarily cover their stuff today, but working with them and doing some, really enabling us to do things we wouldn't be able to do by ourselves. And then certainly, all the different sources of funding the lab has had over the years have been integral in, allowing us to explore this area and, hopefully play a small role in enabling a new class of therapeutics to be available for patients with all kinds of different diseases and injuries, and hopefully, making a positive contribution overall sometime in the near future. So thank you for your time and attention, and we'll be able to take questions now. Thank you.
Questions, I would ask everybody just to unmute themselves, ask a question, but then remember to mute yourselves when your discussion is over so we don't get feedback. Any questions? I have several. It looked as if the lyophilization was significantly better than freezing in the cytokine assay, but just as good in the gap closure assay. And I wonder in the cytokine assay, what do you think that is? My understanding is I have a question about that. Lyophilization, you freeze and then you dehydrate. Do you then store at room temperature or do you store cold?
Yeah, we've stored at room temperature. I think for, a few days. Like I said, we haven't really explored, what could you really do? Can you store this for months or years? We have not done those studies, but you can store it at room temperature at least for a few days after lyophilization, and it does seem to you do seem to be able to reconstitute the EVs and demonstrate that they've retained their functionality in those assays at least. And we use these assays really as just, proxies to let us know if there's been any major changes in what's going on. I think the demonstration that the lyophilized EVs work well in the animal model was some nice to see. I mean, at least, it provides a roadmap for doing further things and kind of real hardcore storage stability testing that would need to be done from a product translation perspective. But yeah, I think the lyophilization is preferable to freezing, but freezing also works just fine, minus 20, minus 80. Again, we haven't really done. I think we have some data about the timing of that out to like a month. Um, but I couldn't tell you, like, if you freeze these things and pull them out after a year, what happens to them. We just don't have those data. But yeah, probably lyophilization is the most promising storage method in our hands.
I also noticed that minus 20 was as good as minus 80 in the gap closure assay, which
Mhm.
seemed useful.
Yeah, I mean, like I said, I think for, these are probably most useful at the moment for, just people doing experiments to know, what you can do, that you can freeze stuff and you know prepare it some days beforehand and freeze it and use it, and it'll still work. Um, and then, like I said, how long you could freeze it, we've done it out to a month and maybe 6 weeks. Um, and we don't see much of a drop off, but I'm sure there is, there's some point in time where it drops off, and we have some samples in the freezer that, we hopefully be able to find out about that at some point in the future.
But so for people, headed for translation or trying to move into humans, your data, you would suggest that they start with lyophilization and start to do that work of seeing how long you can store.
Yeah, I mean, we've certainly seen these discussions around the mRNA vaccines, and how valuable it is to be able to um, have something that can be stored in less stringent conditions. So yeah, I think if it could be lyophilized and stored at room temperature for a long time, that would be great. Um, but even being able to store it at a reasonable temperature in the freezer, not super cold, is still pretty valuable, for the way that, these wouldn't be rolled out and used in the same way as, we're trying to use the vaccines globally in a very, very fast manner.
Sure. Ken Whitwer said he had a question.
I think I might have two. Thanks, Roger, and thanks, Steve, for this really nice talk. I guess I first just wanted to point out that there's been some literature on bacterial OMVs, the outer membrane vesicles, which I didn't really know a whole lot about until we just recently put up a preprint about a bacterial OMV based SARS-CoV-2 vaccination that we tested in hamsters and works pretty well. But there, the first bacterial OMV vaccines were tested back in the 80s. And, so people were lyophilizing these things at least 20 years ago, and found that they were indeed quite stable for quite some time. And especially if you can put them at 4 degrees, but they can even be stable at room temperature. So I think there's a lot of potential there. So that's great. But the question that I had for you was actually more about the functionality. So what does functionality, actually mean? And what do your results tell us about maybe the mode of action of these EVs, like in the wound healing assay or in your animal model. Does it mean, so if you have a fairly destructive or somewhat destructive process where you could have flipping of the topology of some of those proteins that are in the membrane, do you think that the retention of functionality means that that there's a trophic effect mainly of the EVs, or do you think that it's you're at maximum probably getting rid of like half of the normally surface exposed stuff, and so maybe that's just enough to retain a function that would have to do with cargo delivery or signaling?
Yeah, Ken, it's it's obviously a question we're all trying to answer more definitively, and certainly some have taken the stance that the really relatively low cargo to EV ratio with respect to nucleic acids is indicative that that we're probably should be looking mostly for protein mediated phenomena. We really don't know. I mean, I would say our data are somewhat conflicting in this way. I mean, on one hand, we do certainly seem to see effects of loaded cargo, like specifically loaded microRNAs, where, as I was mentioning, like even if you lose a lot of the cargo in a storage process, you still have a lot, and you still have hundreds of copies of a microRNA per EV, and delivering microRNA into a cell, it's not necessarily going to be a linear process, right? You're just going to sort of at some point reach a tipping point where you have enough to regulate the expression of whatever target is downstream of that microRNA. We certainly, in the various different things we've done, like with the protonation method, where you would certainly expect that the surface proteins are significantly disrupted, and yet we don't see differences in uptake. We don't see differences in the immune response or the effectiveness of the EVs in the assays that we do. So that would suggest that maybe the surface proteins aren't playing as strong of a role in that case, and it may be dependent on the type of EV that's being utilized. I still tend to think it's probably a combination of both protein and nucleic acid based effects, but I feel like I've been asking people in the lab for years to just really do definitive studies, get rid of all the proteins and see what happens, get rid of all the RNAs and see what happens, and they're harder to do than to say, and the data never really come back in a completely definitive manner. So, but I think it's definitely something that will really help the field to be definitive about which components of the EVs are really contributing the most to the effects that we see, and then how do we design around that and create therapeutic strategies around that knowledge.
Good point, Steve. Thank you.
H. Wang 4 in chat asks, have you ever checked EV concentration change before and after lyophilization?
Yeah, we've looked at those numbers by NTA, and there's some increase in the NTA counts that we get after lyophilization, and hard to say if that's part of some other settling out of salt particles or other things that are being detected by the NTA apparatus, but it's not a dramatic change. And when we still utilize the NTA numbers to coordinate to normalize for treatments and compare lyophilized versus freshly prepared, we still get similar types of results. So so we've seen some small differences, but nothing that's motivated us to investigate it more thoroughly than that.
Another question from H. Wang 4. You mentioned loading single-stranded but not double-stranded DNA into EVs, does that mean EVs won't take double-stranded DNA?
No, and in fact, I think we had shown double-stranded DNA loading at some point, although maybe I didn't include that slide. But no, it was just just a choice that we made based on the materials that we had and some experiments that we want to do. But no, I mean, there's no reason why double-stranded molecules wouldn't be loaded. I mean, but you would potentially expect the loading to be reduced if you're using one of those diffusion-dependent methods like electroporation or sonoperation, because you just have larger molecules, so reduced diffusion.
We're just about at the hour, but let me ask one more question about your 3D printed perfusion chambers. Once you take the media out of that chamber, do you then purify it in the same way as you would from a flask, like you see as the standard, or is there something about the perfusion chamber that you might get better activity after a less severe purification?
No, the downstream purification is the same. And so that's maybe something that's different depending on how people choose to utilize a hollow fiber bioreactor. So, some of the hollow fiber bioreactor claim is that you can just, by the nature of the fibers, you can just collect EVs directly and not have to do much with them. I'm not so sure that's actually going to be applicable in translational practice, but I don't know for sure. But with the perfusion bioreactor, it's definitely just, you're getting EVs in the media in the same way that you would in a normal flask culture. And so we do TFF and size exclusion chromatography. TFF followed by size exclusion chromatography is our downstream purification method that we think gives us the purest population of EVs. But yeah, so everything you saw there with the bioreactor stuff is also subsequently purified the same way that we did the other things.
Well, great. Really interesting work. Let me tell people that Steve and I ahead of time decided he'd talk about published work so that we can get the video up, since we get some people coming to the video first. So perhaps in a year or two, we can have you come back and talk about the geometries that you're getting in the perfusion chambers and what those do. And we're not going to have an ERCC webinar in August, but in September, Juan Pablo Tosar will be speaking, I believe, about his extracellular ribosome work. So thanks again, Steve, and thanks for a great talk.
Thanks for having me.